Chapter 28 Invertebrates Figure 28.1 Nearly 97% of animal species are invertebrates, including this sea star, Novidina antilensis. Credit: NOAA's National Ocean Service. Introduction: a brief look at any magazine pertaining to our natural world, such as National Geographic, would show a rich variety of vertebrates, especially mammals and birds. To most people, these are the animals that attract our attention. Concentrating on vertebrates, however, gives us a rather biased and limited view of animal diversity, because it ignores nearly 97% of the animal kingdom. The invertebrates, animals that lack a cranium and a defined vertebral column or spine. The invertebrate animal phyla exhibit an enormous variety of cells and tissues adapted for specific purposes, and frequently these tissues are unique to their phyla. These specializations show the range of cellular differentiation possible within the clade Opisthokonta, which has both unicellular and multicellular members. Cellular and structural specializations include cuticles for protection, spines and tiny harpoons for defense, toothy structures for feeding, and wings for flight. An exoskeleton may be adapted for movement or for the attachment of muscles, as in the clams and insects. Secretory cells can produce venom, mucus, or digestive enzymes. The body plans of some phyla, such as those of the mollusks, annelids, arthropods, and echinoderms, have been modified and adapted throughout evolution to produce thousands of different forms. Perhaps you will find it amazing that an enormous number of both aquatic and terrestrial invertebrates, perhaps millions of species, have not yet been scientifically classified. As a result, the phylogenetic relationships among the invertebrates are constantly being updated as new information is collected about the organisms of each phylum. Chapter Outline 28.1 Phylum Porifera 28.2 Phylum Cnidaria 28.3 Superphylum Lophotrochozoa, flatworms, rotifers, and nemerteans 28.4 Superphylum Lophotrochozoa, mollusks and annelids 28.5 Superphylum Ecdysozoa, nematodes and tardigrades 28.6 Superphylum Ecdysozoa, arthropods 28.7 Superphylum Deuterostomia 28.1 Phylum Porifera. By the end of this section, you will be able to do the following: describe the organizational features of the simplest multicellular organisms, explain the various body forms and bodily functions of sponges. As we have seen, the vast majority of invertebrate animals do not possess a defined bony vertebral endoskeleton, or a bony cranium. However, one of the most ancestral groups of deuterostome invertebrates, the Echinodermata, do produce tiny skeletal bones, called ossicles, that make up a true endoskeleton, or internal skeleton, covered by an epidermis. We will start our investigation with the simplest of all the invertebrates, animals sometimes classified within the clade Parazoa, beside the animals. This clade currently includes only the phylum Pachyzoa, containing a single species, Trichoplix adherens, and the phylum Porifera, containing the more familiar sponges, figure 28.2. The split between the Parazoa and the Eumetazoa, all animal clades above Parazoa, likely took place over a billion years ago. We should reiterate here that the Porifera do not possess true tissues that are embryologically homologous to those of all other derived animal groups such as the insects and mammals. This is because they do not create a true gastrula during embryogenesis, and as a result do not produce a true endoderm or ectoderm. But even though they are not considered to have true tissues, they do have specialized cells that perform specific functions like tissues. For example, the external pinacoderm of a sponge acts like our epidermis. Thus, functionally, the poriferans can be said to have tissues. However, these tissues are likely not embryologically homologous to our own. Sponge larvae, e.g., parenchyma and amphiblastula, are flagellated and able to swim. However, adults are non-motile and spend their life attached to a substratum. Since water is vital to sponges for feeding, excretion, and gas exchange, their body structure facilitates the movement of water through the sponge. Various canals, chambers, and cavities enable water to move through the sponge to allow the exchange of food and waste as well as the exchange of gases to nearly all body cells. Figure 28.2 Sponges. Sponges are members of the phylum Porifera, which contains the simplest invertebrates. Credit: Andrew Turner. Morphology of sponges. There are at least 5,000 named species of sponges, likely with thousands more yet to be classified. The morphology of the simplest sponges takes the shape of an irregular cylinder with a large central cavity, the spongocoel, occupying the inside of the cylinder, figure 28.3. Water enters into the spongocoel through numerous pores, or ostia, that create openings in the body wall. Water entering the spongocoel is expelled via a large common opening called the osculum. However, we should note that sponges exhibit a range of diversity in body forms, including variations in the size and shape of the spongocoel, as well as the number 724 Chapter 28, Invertebrates access for free at openstax.org. And arrangement of feeding chambers within the body wall. In some sponges, multiple feeding chambers open off of a central spongocoel, and in others, several feeding chambers connecting to one another may lie between the entry pores and the spongocoel. While sponges do not exhibit true tissue layer organization, they do have a number of functional tissues, composed of different cell types specialized for distinct functions. For example, epithelial-like cells called pinacocytes form the outermost body, called a pinacoderm, that serves a protective function similar to that of our epidermis. Scattered among the pinacoderm are the ostia that allow entry of water into the body of the sponge. These pores have given the sponges their phylum name Porifera, porebearers. In some sponges, ostia are formed by porocytes, single tube-shaped cells that act as valves to regulate the flow of water into the spongocoel. In other sponges, ostia are formed by folds in the body wall of the sponge. Between the outer layer and the feeding chambers of the sponge is a jelly-like substance called the mesoil, which contains collagenous fibers. Various cell types reside within the mesoil, including amoebocytes, the stem cells of sponges, and sclerocytes, which produce skeletal materials. The gel-like consistency of mesoil acts like an endoskeleton and maintains the tubular morphology of sponges. The feeding chambers inside the sponge are lined by choanocytes, collar cells. The structure of a choanocyte is critical to its function, which is to generate a directed water current through the sponge and to trap and ingest microscopic food particles by phagocytosis. These feeding cells are similar in appearance to unicellular choanoflagellates, protista. This similarity suggests that sponges and choanoflagellates are closely related and likely share common ancestry. The body of the choanocyte is embedded in mesoil and contains all the organelles required for normal cell function. Protruding into the open space, inside the feeding chamber is a mesh-like collar composed of microvilli with a single flagellum in the center of the column. The beating of the flagella from all choanocytes draws water into the sponge through the numerous ostia, into the spaces lined by choanocytes, and eventually out through the osculum, or osculi, if the sponge consists of a colony of attached sponges. Food particles, including waterborne bacteria and unicellular organisms such as algae and various animal-like protists, are trapped by the sieve-like collar of the choanocytes, slide down toward the body of the cell, and are ingested by phagocytosis. Choanocytes also serve another surprising function. They can differentiate into sperm for sexual reproduction, at which time they become dislodged from the mesoil and leave the sponge with expelled water through the osculum. Link to learning. Watch this video to see the movement of water through the sponge body. Click to view content, https://www.openstax.org/l/filter_sponges. The amoebocytes, derived from stem cell-like archaeocytes, are so named because they move throughout the mesoil in an amoeba-like fashion. They have a variety of functions. In addition to delivering nutrients from choanocytes to other cells within the sponge, they also give rise to eggs for sexual reproduction. The eggs remain in the mesoil, whereas the sperm cells are released into the water. The amoebocytes can differentiate into other cell types of the sponge, such as collinocytes and lophocytes, which produce the collagen-like protein that support the mesoil. Amoebocytes can also give rise to sclerocytes, which produce spicules, skeletal spikes of silica or calcium carbonate, in some sponges, and spongocytes, which produce the protein spongin in the majority of sponges. These different cell types in sponges are shown in Figure 28.3. 28.1. Phylum Porifera 725 Visual connection. Figure 28.3 Simple sponge body plan and cell types. The sponges, a. Basic body plan and, b. Some of the specialized cell types found in sponges are shown. Which of the following statements is false? A. Choanocytes have flagella that propel water through the body. B. Pinacocytes can transform into any cell type. C. Lophocytes secrete collagen. D. Porocytes control the flow of water through pores in the sponge body. Link to learning taken up close tour, http://openstax.org/l/sponge_ride through the sponge and its cells. As we've seen, most sponges are supported by small bone-like spicules, usually tiny pointed structures made of calcium carbonate or silica, in the mesosole. Spicules provide support for the body of the sponge, and may also deter predation. The presence and composition of spicules form the basis for differentiating 3 of the 4 classes of sponges, figure 28.4. Sponges in class Calcarea produce calcium carbonate spicules and no spongin. Those in class Hexactinellida produce 6-rayed siliceous, glassy, spicules and no spongin, and those in class Demaspongia contain spongin and may or may not have spicules. If present, those spicules are siliceous. Sponges in this last class have been used as bath sponges. Spicules are most conspicuously present in the glass sponges, class Hexactinellida. Some of the spicules may attain gigantic proportions. For example, relative to typical glass sponge spicules, whose size generally ranges from 3 to 10 mm, some of the basal spicules of the Hexactinellid monorifice chuni are enormous and grow up to 3 meters long. The glass sponges are also unusual in that most of their body cells are fused together to form a multinucleate syncytium. Because their cells are interconnected in this way, the Hexactinellid sponges have no mesosole. A 4th class of sponges, the Sclerospongiae, was described from species discovered in underwater tunnels. These are also called coralline sponges after their multi-layered calcium carbonate skeletons. Dating based on the rate of deposition of the skeletal layers suggests that some of these sponges are hundreds of years old. 726 Chapter 28, Invertebrates access for free at openstax.org. Figure 28.4 Several classes of sponges. A. Clathrina clathris belongs to class Calcarea. B. Staurocalyptus spp. Common name, yellow Picasso sponge, belongs to class Hexactinellida, and C. Acanos erythicus belongs to class Demaspongia. Credit A. Modification of work by parent Jerry. Credit B. Modification of work by Monterey Bay Aquarium Research Institute, NOAA. Credit C. Modification of work by Sanctuary Integrated Monitoring Network, Monterey Bay National Marine Sanctuary, NOAA. Link to learning use the interactive sponge guide, http://openstax.org/l/id_sponges to identify species of sponges based on their external form, mineral skeleton, fiber, and skeletal architecture. Physiological processes in sponges. Sponges, despite being simple organisms, regulate their different physiological processes through a variety of mechanisms. These processes regulate their metabolism, reproduction, and locomotion. Digestion. Sponges lack complex digestive, respiratory, circulatory, and nervous systems. Their food is trapped as water passes through the ostia and out through the osculum. Bacteria smaller than 0.5 microns in size are trapped by choanocytes, which are the principal cells engaged in feeding, and are ingested by phagocytosis. However, particles that are larger than the ostia may be phagocytized at the sponge's surface by pinacocytes. In some sponges, amoebocytes transport food from cells that have ingested food particles to those that do not. In sponges, in spite of what looks like a large digestive cavity, all digestion is intracellular. The limit of this type of digestion is that food particles must be smaller than individual sponge cells. All other major body functions in the sponge, gas exchange, circulation, excretion, are performed by diffusion between the cells that line the openings within the sponge and the water that is passing through those openings. All cell types within the sponge obtain oxygen from water through diffusion. Likewise, carbon dioxide is released into seawater by diffusion. In addition, nitrogenous waste produced as a byproduct of protein metabolism is excreted via diffusion by individual cells into the water as it passes through the sponge. Some sponges host green algae or cyanobacteria as endosymbionts within archaeocytes and other cells. It may be a surprise to learn that there are nearly 150 species of carnivorous sponges, which feed primarily on tiny crustaceans, snaring them through sticky threads or hooked spicules. Although there is no specialized nervous system in sponges, there is intercellular communication that can regulate events like contraction of the sponge's body or the activity of the choanocytes. Reproduction: sponges reproduce by sexual as well as asexual methods. The typical means of asexual reproduction is either fragmentation. During this process, a piece of the sponge breaks off, settles on a new substrate, and develops into a new individual, or budding. A genetically identical outgrowth grows from the parent and eventually detaches or remains attached to form a colony. An atypical type of asexual reproduction is found only in freshwater sponges and occurs through the formation of gemmules. Gemmules are environmentally resistant structures produced by adult sponges, e.g., in the freshwater. Spongilla. In gemmules, an inner layer of archaeocytes, amoebocytes, is surrounded by a pneumatic cellular layer that may be reinforced with spicules. In freshwater sponges, gemmules may survive hostile environmental conditions like changes in temperature, and then serve to recolonize the habitat once environmental conditions improve and stabilize. Gemmules are capable of attaching to a substratum and generating a new sponge. Since gemmules can withstand harsh environments, are resistant to desiccation, and 28.1, phylum Porifera 727 remain dormant for long periods, they are an excellent means of colonization for a sessile organism. Sexual reproduction in sponges occurs when gametes are generated. Oocytes arise by the differentiation of amoebocytes and are retained within the spongocoel, whereas spermatozoa result from the differentiation of choanocytes and are ejected via the osculum. Sponges are monoecious, hermaphroditic, which means that one individual can produce both gametes, eggs and sperm, simultaneously. In some sponges, production of gametes may occur throughout the year, whereas other sponges may show sexual cycles depending upon water temperature. Sponges may also become sequentially hermaphroditic, producing oocytes first and spermatozoa later. This temporal separation of gametes produced by the same sponge helps to encourage cross-fertilization and genetic diversity. Spermatozoa carried along by water currents can fertilize the oocytes born in the mesoel of other sponges. Early larval development occurs within the sponge, and free-swimming larvae, such as flagellated parenchyma, are then released via the osculum. Locomotion sponges are generally sessile as adults and spend their lives attached to a fixed substratum. They do not show movement over large distances like other free-swimming marine invertebrates. However, sponge cells are capable of creeping along substrata via organizational plasticity, i.e., rearranging their cells. Under experimental conditions, researchers have shown that sponge cells spread on a physical support demonstrate a leading edge for directed movement. It has been speculated that this localized creeping movement may help sponges adjust to microenvironments near the point of attachment. It must be noted, however, that this pattern of movement has been documented in laboratories. It remains to be observed in natural sponge habitats. Link to learning. Watch this BBC video, http://openstax.org/l/c_sponges showing the array of sponges seen along the Cayman Wall during a submersible dive.